Okay. All right. God, such a pain in the ass. Be with you in just a second. Trying to sort something out. Okay, so essentially, today is two things to talk about. One is any questions any of you may have about producing that graph that I'm after for tonight's assignment for due today by midnight. And the other, sorry, the other thing to talk about is the mini lab report for next Tuesday. Actually, I might actually extend that to Thursday, thinking about because we've got an exam on Tuesday, and I try not to have due dates on the same day because that's a little rude. Let's do that. I never get complaints about extending deadlines. Um, that's for sure. Uh, yes, the mini lab report next week. Let's make it. What date is that? 9th. Yeah, sometimes I don't double check between um, lecture and lab. Sometimes there's some conflicts which aren't intended. So in response to Olivia and Alyssa, yes, it's just the graph. So in an Excel spreadsheet. And so the reason why I like to have the spreadsheet is because then I can see, oh yeah, correct. Um, yeah, so it's just the graph, but if you send me the spreadsheet, then I can see the table and the process. Essentially, you need to follow the instructions that Dr. Page lays out in that video, which wouldn't hurt to kind of point it up again, just in case you missed it the first time around. Here we go. And use the data in the lab handout, which is, I'm not sure if this will actually paste, but it's just above the, yeah, that looks like ass. Yeah, you can figure it out though. So hang on, Angela, let me just make sure I've answered everyone's questions first before we move on. Yeah, just use those data. You need to work out, basically sum up parentals and recombinants and express those in a graph with what you would expect if there is no linkage, right? So if you follow through that video, it's like half an hour, but you can skip ahead if you know how to do various bits of it. Essentially, you'll end up with a nice little bar graph with 4 different bars on it: parentals, recombinants, observed, expected for both. And that's really it. It's not a lot. And yeah, it's in the table under data analysis. It's the same one that I just copied and pasted up there. And the pluses as well for the lab report that you need to do for next Thursday, you will be using that graph. So you've already done a big chunk of the results section of the mini lab report anyway. Okay, so Angela's question. Yeah, let's go back here. So the whole series of modules, which is essentially the last kind of half of the lab, is going to be starting next Tuesday, but the first deadline for that is the following Tuesday. Give me just a second, Sky. And that will be the, yeah, DNA isolation. So I haven't actually figured out how to do that yet, because that's going to be a new thing being made. I think COVID happened just after that last semester. That assignment will be done in the next week or two. And then when it is ready, it will be in the same folder as you will find that particular module, as we have done for all this other stuff. Yeah, it was interesting, that's for sure. Okay, to Sky. So no, you do not need the table or figure legends in the Excel spreadsheet, just in the report. Yeah, because essentially, yeah, you're doing the analysis. So what you handing in today is the data analysis, at least the initial part of it. And then you're going to finish that up with a little bit of statistical analysis, the chi-squared test and odds ratio. And then you're going to kind of fit that into the bigger picture with the lab report. So essentially you can do that, and this is in the answer to a question as well. You can hand that in at any point, right? And as soon as you're happy with it, I can offer you feedback on it too. But as soon as you're happy with it and you consider it done for the lab report, then you're good. So you don't need to hand anything else in if you've already done this work already. I'm trying to keep that non-specific. Right. I haven't actually had a chance to look at any stuff that people have sent me over the last few days. It's been pretty hectic. But, geez, what day is it today, actually? Thursday. Yesterday wasn't hectic. That was nice. Yesterday I just fixed a whole bunch of solar lamps for the garden and played tennis. But anyway, if you send me stuff, then I've received it, and as long as you're happy with it as it stands, you're good. If you want feedback on any of it, then just let me know, and I'll give you that too. But I'll actually, while I'm thinking about it, because I don't want to forget, and only half of the class are here. Let's just, I'll get an announcement up so I don't forget to send out about the deadline change for the mini lab report. I'll be back in just a second. I just want to create this and then I'll finish off after lab. Was it 9th? Let's just check that. Yeah, that's right. Yeah, no worries. Okay, so those are some good questions. I'll send that announcement about the deadline extension in a little bit. I just got it up so that I don't forget to do it, essentially. Sometimes when I finish these, I'm like, feel like I'm dead, so I don't always get onto. I've got other stuff like the YouTube videos and things to do. I don't always remember stuff. Okay, so does anybody have any questions about the any more questions rather about the graph and Excel spreadsheet for tonight? Especially how to do it, if anyone has any hiccups in like the actual process itself. Doom, doom, doom. Yeah, awesome. Okay, so yeah, I thought he did a fabulous job, to be honest. He's pretty much who you would have got for genetics uh, if I hadn't been teaching it. You know, so he was the genetics teacher here for the last 3 years, 4 years, and now he's doing biostatistics, and so I'm pretty much taking over genetics from him. So that's why a lot of the stuff is kind of in transition from one to the other, like the PowerPoints, for example. Okay, so in terms of the lab report, and I do again encourage you to seek feedback, either from other people or from me. Probably from me will be, you know, perhaps more useful, maybe because I know exactly what I'm looking for. But certainly get some feedback on your lab report because then you can improve it by a considerable amount, right? And this is part of the process of doing this, right? Feedback is actually part of that. And it certainly is in anything I've ever written. I've never, ever written something in one go and then submitted it and got it accepted. It just doesn't work that way. Um, so essentially, what I'm after is this will take a little while. Mini introduction, because it's a mini lab report. Um, bu- bu- bum. Also, before I forget, use headers. Like, God damn it, can I not type? Introduction, methods, results, discussion. That's not just, well, that's for a bunch of benefits. One is I know what I'm reading about, because it says methods. So, oh yeah. And so I go, okay, we're reading about methods now. Great. Here's the grade for methods. Or even just, oh, now they're talking about what they did, right? Rather than what they're going to do, which is really introduction. So that is important. It helps me figure out what exactly I'm reading about at a given point. But arguably the most important part is it helps you think clearly about what you're writing. Because one of the most common things that happens in scientific writing for undergraduates, at least earlier on, is smooshing stuff together. So essentially, we'll have one giant paragraph, which is introduction, and then a bit of methods, then a bit of introduction, and then some results, and then a bit of discussion, then maybe some methods, and then back to results. Yeah, not headers on each page. So yeah, sorry, not headers and footers in like the Word document sense, but section headers. I forgot that there's more than one meaning for the word headers. So you separate each section of the report into different pieces based on its purpose. And so by putting in introduction, and actually I personally do this as well, I put in those headers before I do any other writing. I'll put in the introduction, methods, results, abstract for like fancier stuff, references, acknowledgements, stuff like that. I put that in first because they're like placeholders. It's like, I need to do the introduction. And sometimes I don't want to do no introduction no more. I'm going to do some methods today instead and just kind of trundle away at that. For this particular project, you don't need subheaders because you're only really doing one thing, right? But if you were doing multiple experiments and you're turning it into a manuscript, each different approach would have its own subheader. So, like in my work, worm maintenance, RNAi, construct, transgene construction, whatever, phenotyping, will all have their different subheaders because that's where those different blocks of information go. And so really for your benefit, and mine as well, because I have to read them, these headers and subheaders, if you need them, or section headers, help organize your thoughts, right? And it's quite clear that if you start describing the cross in your introduction, it's like, whoa, whoa, whoa. That's methods, right? You can describe that you will be doing a cross with the white and yellow genes, but you can't describe what the cross is that you did because that's methods, right? And so it kind of helps you put different bits of information into different places, right? That's the key benefit of using section headers. All right, methods. Again, brief, short, whatever word you want to use. So essentially, this is a short description of the back cross that you did, right? Obviously, you didn't do it, but you would have if you'd been in face-to-face labs. So what did you do? I've laid it out hopefully quite clearly for you in the lab handout. I want you to essentially simplify and paraphrase that so that if I were to read it from scratch, or if, say, I sent one of these to Dr. Page, he'd be able to go, oh, that's what they did, right? Fairly straightforward. And then a brief description of the statistical analysis. So what tests? Right? Again, oh, I forgot this. Shouldn't be more. So again, no need to laboriously kind of type out all of the different method steps that were described in the handout, because I wrote that. I don't need to see that anymore. What I want to see is that you're able to understand it sufficiently to kind of condense it down into a shorter, more manageable description. Right? So essentially there's what you did, which was the cross, and how you analyzed it, which is the chi-squared and the odds ratio, and also recombination frequency. Oh. So in many ways, and I actually tell this to my kids when they're writing an essay or something, you can break this down into, I'm going to tell you what I'm going to do. I'm going to tell you what I did. I'm going to tell you what I found. I'm going to tell you what it means, right? That's your introduction, what I'm going to do. Methods, what I actually did. Results, what did I find? Discussion, what does it mean? Note these two. Results is what I found, not what it means. Discussion is what it means, not what you found. Kind of. It's a little bit trickier, right? But you shouldn't be discussing your results in the results section. You just describe them. Discussion is where you discuss your results. All right, results. I might have to check my lab handout to make sure I don't forget anything here. So results are the big bit. That's what matters. So you need have You can also put the table and table legend into if you wish. Yeah, I'm not going to ding you particularly hard if it isn't there, but it's not going to hurt you if you do put it there. Let's put it that way. You need your chi-square, oops, wrong symbol. So you need your value, df, p-value. Odds ratio. And recombination, oops. And map distance between yellow and white. Let me just double check, make sure I haven't forgotten anything else. Oh yeah, I did. Hang on a sec, Olivia. So this is an important thing which students often forget. They often think that, okay, I've put a nice graph in here, it's got a figure legend, put in some like statistical analysis, I'm good. And so imagine if you were reading this, you would be presented with a graph and some stat stuff. How would you know what it is that you're looking at? I mean, the figure legend will help, but how does that relate to the table? right, that is also there. So you need some text describing what it is you have in your results section. If you don't have that, you cannot get full grade for the results section, even if you put everything else in, right? There needs to be a little bit of text, not a lot, but a little bit to cover that, right? So your results in total should probably be about a page, would be my guess. About half a page for the graph, maybe quarter of a page for a table, maybe another half page for a table, doesn't really matter. And then, kind of like that much for the description of your results. So to answer Olivia's question, in total, lab report should be about 2 to maybe pages. I'm not going to be grading you on length, right? What I'm going to be looking for is, have you done all of these different things? If you have, you get full marks, right? If you're missing anything out, then most likely you won't get full marks. You'll get deducted a bit for whatever you're missing. I'm also not going to be ragging you about grammar and sentence structure and things like that, because to make that worthwhile, I would need to give you time to respond to that feedback. And because of the way we're doing it, that's not going to be the case this semester. So ideally, it's going to be legible, right? I'm going to understand what you're saying, and there aren't going to be too many typos and errors because that just looks bad, right? So do try and do a decent job of making it look the way you want it to look. But in total, 2.5 pages should be sufficient to cover all of these different points. Okay. And then discussion. And this is actually, discussions are often the hardest part to write, to be honest. And so, short recap of results. Which is essentially going to be your chi-square, odds ratio, and map distance, and what it means. As basically, are these two genes linked or not? And if so, how closely? That's really it. Do all that, you'll get full grade. I'm not going to be going through there trying to pick things apart. I'm actually going to be going through trying to give you points wherever I can, is generally the approach I take to grading stuff like this. So in terms of writing it, what I would suggest, and I actually do this again myself. Oh, I wrote that. It's like, that's a weird thing to ask. Is kind of even copy these things down, right? I'm going to post this chat up to Blackboard as soon as I'm done with this, along with like the YouTube video to YouTube. But what I find actually helps a lot is to copy these things down, right? So copy and paste the introduction and then separate it into the different things like bullet points. Copy and paste the results. This is what I need to do. Ding, 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 ding. All of these things. And a discussion. And then you simply just fill in the bullet points and turn them into sentences and paragraphs. That's the easiest way to do this, right? You're not starting from scratch. I've already given you the whole structure and essentially all the different things you need to do. You just need to put that together in a coherent format and make sure you do each of those different things. If you do all of them, then you get the full grade. Or I mean, obviously you have to do them all well. If you put in like a graph drawn with crayon, and smiley faces and stuff, you might not get full points for the graph part. But anyway, you get the idea. Okay. Anybody got any questions? And this is also worth quite a chunk. I can't get to my get that back. Let me just see. I can't remember off the top of my head. It's down here somewhere. All right, so the spreadsheet is 100 points and the mini lab report is 200 points. And so in total, these two things, which are essentially paired together, is I think 300 points out of 700. Yeah. So Excel spreadsheet, graph, and lab report. Points for lab. So, oh man, I got to do some math. That's hard. Use a calculator. It's always the best option. So that's 43% of your lab grade, which is 10% of your total course grade overall, which, is a reasonable chunk. So it pays to spend a bit of time and do a good job. And so just as a reminder, yeah, due this time next week. So if you wish to, you can actually spend time focusing on your exam, which is a really big chunk of your grade, and then kick into action on this after you've done the exam. So you still have all of Wednesday and, obviously most of Thursday to do it. Okay, questions. Now's a good time. And if any of you have submitted a lab report already and you wish to make changes to it, that's totally fine as well. Just let me know so I mean, technically speaking, I'm not going to be grading these lab reports until not this weekend, probably the weekend following, right? Maybe a week tomorrow, maybe Friday the 10th, depending on what I've got going on elsewhere. So you have until then to change your mind. If you want to go back and look through it or you want more feedback, that is totally fine as well. But please do make the most of the opportunity to get feedback. It will make your lab report better and you'll get more points. Yeah, that's totally fine. Again, it's not bad. It's just a part of the learning process. But it also one thing worth reiterating, and it's still a tricky thing for people who've been doing this for a while, is to distinguish between results and discussion. And so results is only ever a statement of what you found. If you ever feel like talking about what it means, stop. That's the discussion. So, for example, if your chi-square result, you found the p-value is really low. So we got chi-squared, 55, df 7, p-value of 0.0001, right? If you find yourself typing, and that means, stop. That's discussion. Now, in the discussion, what you can do is you go, we found that our null hypothesis that no linkage was occurring was rejected because we got a highly significant p-value from the chi-squared test. This means that these two genes are linked. That's discussion, right? So hopefully you can see talking about interpreting results or what it means or how it fits into a bigger picture, that's always discussion, because the results is where you actually present your results. It's a tricky thing to get. It's quite common to end up discussing things in results, but if you find yourself doing that, stop. Copy and paste that sentence into the discussion section, because that's where it belongs. And also another, from terms of grading point of view, if I see discussion stuff in the results, I'm not going to ding you for it in the results, but I'm also not going to give you that part of the grade for the discussion if that isn't in the discussion, right? So I'm looking for specific things in specific places. If you do all of your discussion in the results section, then there's just one sentence in the discussion, I'm only going to be grading your discussion part on that one sentence. All the other stuff that's elsewhere, you're not going to get the grade for, right? And that would be a pain because you've done the work, right? You've already done the thinking and the writing. You just not put it in the right place. So again, remember, put the interpretation or meaning of stuff in the discussion, because that's where I'm going to be looking for it. And I don't want to not give you the points, but I am only going to be grading you each section for what should be in that section. Does that make sense? Cool. All right. Last call for any questions or comments. Okie dokie. So again, please do send me your initial versions. I'm not going to say final versions because they won't be final until I've given you some feedback. Right? You should be doing at least 2 or 3 drafts before you finish your final draft for submission. And even then, there may be things you might want to change. Okay, so please do make the most of that. It will help. All right. Well, thanks very much, everybody. Hope you all have a fabulous loss of the Empire celebrations on the 4th. I shall be wearing black and in mourning, obviously, because that's what we do in the UK. That's just a joke. We don't even know what 4th of July means in the UK, to be honest.